Hello friends, Ace Botany next episode. In this episode, we'll be discussing Chapter 6, Cell, the Unit of Life, second part. And what all things we'll be discussing in this episode? As usual, for that, I will share the screen so it will be convenient for you. I think the screen came, so we can proceed. So Chapter 8, Cell, the Unit of Life, Episode 2, topics. As usual, there will be an introduction. Then prokaryotes will be expanding in this episode from cell envelope and its modification. Then ribosome, inclusion bodies, eukaryotic cell, just an introduction only. Then as usual, 5 theory question, 5 NEET model question with the key. Remember, NEET 26 for that. Daily questions I started to put on YouTube. That is in Ace Botany. So subscribe it. So you'll be getting this type of classes as well as notes and questions for NEET exam. I think it is clear, so we can proceed. So as an introduction, last episode, we observed cell is the basic structural, functional, fundamental unit of life. And cell, since it is the basic unit of life, it has to be protected. And there will be many coverings, specifically when we come to prokaryote, which include, there is a glycocalyx, cell wall, cell membrane, and its modifications. There is ribosome, inclusion bodies. In prokaryotic cell, we can specifically observe. Then eukaryotic cell introduction, questions as usual. I think introduction is clear. Now we can proceed to prokaryote remaining topics. So prokaryotic cells, cell envelope and its modifications. Most prokaryotic cells, specifically bacterial cell, that is our main item, bacterial cell, have a chemically complex cell envelope. It is tightly bounded by 3-layered structure. It's important. Which are these 3-layered structures? That is outer glycocalyx, second is cell wall, and third is plasma membrane or cell membrane. And remember, each layer has got its own functions to perform. I think first page is clear. We can proceed to second page. I think second page came. Now we are entering into prokaryotic cell from outer, that is glycocalyx. What are the features of glycocalyx? It is usually outermost layer, a coating with a mucus or polysaccharide. Protection of cell is the main purpose. Also helps in adhesion. In different bacteria, this layer varies in thickness and chemical composition. So it differs. If it is loose, then we'll be calling it a slime layer. And major function of the slime layer, that is, prevents the loss of water and nutrients from cell. This type of question common for NEET exam. If it is thick and tough, then the term what we use is capsule, giving gummy or sticky nature to the cell. Similarly, it also helps to hide from host immune system, important for exam. I think that 2-layer clear. Now, one more point regarding capsule and slime layer. Basically, polysaccharide, sometimes proteins also seen. I think idea clear. Now we can enter into second layer, that is cell wall. Cell wall is the structure which determines the shape of the cell. Important question. Now, what else cell wall provides? It can also provide a structural support by which bursting of bacterium and collapsing of bacterium is prevented because it may be more rigid. Chemically, it is made by peptidoglycan, important. Fungal cell wall, bacterial cell wall, then plant cell wall, that way, different chemicals. So chemically, it is made up of peptidoglycan. So what is this peptidoglycan composition? It is having N-acetyl glucosamine, short form is NAG, and N-acetyl muramic acid, short form is NAM, and they're linked by beta-1,4 glycosidic bond. It is always called murin or mucopeptides. Antibiotics able to digest peptidoglycan linkages can act against those bacteria with the peptidoglycan cell wall. I think that idea is clear because when we discuss mycoplasma, you can find this wall-less bacteria where these antibiotics will not work because peptidoglycan cell wall we cannot find. When we discuss mycoplasma, I will repeat it. This important question in that area. I think idea clear. Now comes staining of bacteria. Stain means color or color imparting substance. The most famous scientist involved in bacterial staining is Christian Gram, a Denmark or Danish botanist. Lifespan 1853 to 1938. And from its classification, bacteria classified into 2 groups. One is gram-positive, that means which will retain the stain. One is gram-negative, that is not able to retain stain. Basic steps involved. 5-6 steps are needed. Microbes also, we may be needed this in second year. Basic steps involved. 1. Take bacterial assembly. 2. Just dry it. 3. Stain with the crystal violet, which is otherwise called gentian violet. So crystal violet and gentian violet are same. So all bacteria will be getting color of the stain. Now wash with water. Treat with iodine solution. That is an area of question. Iodine solution will act as a mordant. Mordant in chemistry may be studied, usually attaches the dye firmly to the structure, otherwise often called as dry fixative, because dye will be firmly attached with the help of mordant. Here it is iodine solution. And that is due to formation of a chemical complex. Then next step is treating with alcohol. So usually it acts as a decolorizer. Those who retain violet after treating with the alcohol, they are called gram-positive. And those who lost the color, counterstain with the safranin, become pink, it is called gram-negative. I think idea clear. Stepwise procedure. Some examples shown below for gram-positive and gram-negative bacteria, a question can be in 26. Gram-positive, Staphylococcus, many are there. Corynebacterium, Clostridium, Lactobacillus. Gram-negative, Xanthomonas, Vibrio, Salmonella, Acetobacter, and an important specimen is E. coli. It is a symbiotic bacteria in our intestine. It is gram-negative. And why this permanent staining comes? It is due to many reasons. Many reasons that permanent staining can be there. So 2 examples are given there. One is thickness of cell wall. Second is murin content amount. So in both cases, more in gram-positive bacteria, still many other factors are there to get gram-positive and gram-negative features. I think idea clear. Now we can proceed to next area. Next is plasma membrane. Features. It is selectively permeable. It originally interacts with the outside. It is structurally similar to that of eukaryote. So glycocalyx, cell wall, plasma membrane. Now comes mesosome. It is a special membranous structure. Mesosome, it is formed by the invagination of plasma membrane into the cell. Here you can find it. Present NCERT, no such diagram directly available. That is why I put this diagram from old NCERT. Both the diagrams, it is from old NCERT. In page number 90 of present NCERT, there is a diagram, but marking is not there. That is why I put this diagram. This one, I think it is clear. Now, this is different shapes of bacteria, because when I search old NCERT book, it's with me, so I am able to look at this type of diagram. So very useful for you. Only rough reference, because present NCERT, this type of diagram is not available. That is why I wrote the heading old NCERT figures just for a rough reference. So it'll be easy for you. I think idea clear. Now we can continue. That is mesosome. So mesosome can exist in 3 forms. Which are what are 3 forms? Famous question. One is vesicles, second is tubules, and third is lamellae. 3 forms it can exist. Now, as usual, functions of mesosome. So mesosome connected with the cell wall formation, direct NCERT important. DNA replication and distribution to daughter cells. Helps in respiration like mitochondria. Mesosome in prokaryotes. Then secretion process to increase the surface area of the plasma membrane and enzymatic content. Next is an important one. Mesosome is common in gram-positive bacteria. So mesosome common in gram-positive bacteria. So many NEET type questions we can anticipate. Many points are there. Maximum I put a star mark to give extra importance. In some cyanobacteria, purple bacteria, we can find some other membrane extensions also seen into the cytoplasm, which is often called, term you may be familiar, chromatophores. Phore means stalk, chromo means color. That means pigments bearing structural pigments seen. Pigments means helps in photosynthesis. I think idea clear. Now we can proceed to motility of bacteria. Bacterial cells may be motile or non-motile. If motile, it is linked with the flagella. Cilia not seen important. So motility is linked with the flagella. Number of flagella variable, one side, both side. That way different classifications are there. Monotrichous, peritrichous, lopotrichous, amphitrichous. Because it is not coming into the syllabus, I just number of flagella variable by which I just close that area. Now comes structure of flagella. Structure of flagellum. See, bacterial flagellum consists of 3 parts. From top, diagram given this also old NCERT diagram, but it's clear. From top means from outside, it is filament. Then this is the hook area. Then bottom part is basal body. So diagram, since it is there, in old NCERT, it will be clear for you. So filament, hook, and basal body. Filament extends from cell surface to outside. That is hair-like structure, what we observe outside. It is the longest part, cell surface to outside. And chemically, it is by a protein called flagellin. In eukaryote, it is tubulin. When we come to eukaryotic flagella, I will explain there. So bacteria, it is flagellin. Basal body structurally have a rod-like structure. See, this is the rod. I marked there. Rod-like structure on which rings will be arranged. Say rings 1, 2, 3, 4. 4 rings will be arranged. Maximum marked there, so it will be easy for you. In gram-positive bacteria, 2 rings, SM. In gram-negative bacteria, 4 rings. Can be a question even though not directly mentioned in NCERT. Bacteria, 4 rings, LPSM. Rings M and S in cell membrane. So everything marked there. So this is cell membrane. And rings PL, that is in cell wall. I think I did clear that the rings, everything clear. Old NCERT diagram is very clear. So we can proceed to next area. Next structure for us to discuss is pili and fimbriae. So besides flagella is already there. Pili, fimbriae are also surface structures seen in bacteria, but not connected with the motility. So what is pili? What is fimbriae? What is the chemical nature? What is the function important for exam? Pili is elongated, tubular, made by special protein called pilin, connected with the sexual reproduction. Up to that, I think it is clear. Now, where we can find true pili, like a mesosome. So true pili is commonly seen in gram-negative bacteria. Sex pili is another name. F pili is another name, synonym. Fimbriae. Fimbriae are small bristle-like fibers sprouting out of the cell. Function in some bacteria, they are known to help in attachment of bacteria to rocks in streams and host tissues. This is the diagram for pili. Again, old NCERT diagram, presently not there. Pili is different, flagella is different. Next, ribosome and inclusion body. In prokaryote also, we can find ribosome, and ribosome associated with the plasma membrane. Size is 50 nanometer by 20 nanometer. And made by 2 subunits, like eukaryotic ribosome, 50S and 30S. Eukaryotic is 60 and 40. There we can study later. So 50 and 30, together 70S. Main function, it is the site of protein synthesis as usual. And several ribosomes may attach to single mRNA, forms a chain called polyribosome or polysome. There is one more synonym for it. Not commonly seen, it is also called ergosome. So mainly polyribosome or polysome. One more term that is not so common, it is ergosome. Ribosome of a polysome translate mRNA into protein. So protein synthesis. I think idea clear. Now we can proceed to next structure, that is inclusion bodies. Inclusion bodies. What are the features? A regular question, theory and run. All connected with NCERT. Reserve material in prokaryotes. Then it is stored in the cytoplasm. It is in the form of inclusion bodies, reserve material. They are not bounded by any membrane system. They lie free in cytoplasm. Typical example, 2 sets are there. One is phosphate granule, synophysin granule, glycogen granule. Remember, is a wet PCG. Now, another set, there are another type of inclusion bodies. There may be a covering, usually single layer, but it is non-unit membrane and 2 to 4 nanometer thick. An example for it is sulfur granules, gas vacuoles, poly beta hydroxybutyrate granules. Again, PSG. Gas vacuoles are seen in blue-green algae, purple and green photosynthetic bacteria. NCERT by number 91 all are given. I think outside here. Now, next is eukaryotic cell and introduction. So, prokaryote is getting over. See, whenever the term EU comes, EU came from Greek word. EU came from Greek word, or Greek means true, good, etc. That is corresponding meaning. EU came from Greek, means true or good. Karyo means nucleus, familiar to us. In totality, eukaryotic cell means cells with organized nucleus and nuclear envelope. That's the meaning of the term eukaryote that we'll be continuing in next episode. And there will be 5 theory questions. And you can find 5 NEET model question also with the key. That is regarding the second episode of cell, the basic unit of life, that is chapter 8. as a totality, introduction, prokaryotic cell envelopment modification, ribosome inclusion body, eukaryotic cells, introduction question. And introduction, we just entered into the topic. What all things we'll be learning, like glycocalyx, cell wall, cell membrane modification, ribosome, etc. Then cell envelope and its modification. So most prokaryotic cells, specifically bacterial cell, have a chemically complex cell envelope. It is tightly bound by three-layered structure, important, glycocalyx, cell wall, plasma membrane. Then glycocalyx, what are the features? If it is loosely arranged, slime layer prevents the loss of water and nutrients from cell. If it is thick and tough, it is capsule. Capsule, giving gummy sticky nature to the cell, helps to hide from host immune system. And chemically, polysaccharide, both of this, and sometimes protein present. Then we enter into cell wall. Cell wall is structure which determines the shape of the cell. It also provides structural support by which bursting of bacterium and collapsing of bacterium is prevented. Chemically, peptidoglycol, NAG-NAM linkage, that is linked by beta-1,4 glycosidic bond, murin or mucopeptide, antibiotic, which is able to digest peptidoglycan linkages. In those cases only, we can give specific antibiotic to destroy the infection, because that antibiotic will work on peptidoglycan cell. Otherwise, no use. Staining of bacteria. It is concerned with the famous scientist Christian Gram, Danish botanist. Very old method, still it is utilized. Then those who retain violet coloration, gram positive. Those who lost, gram negative. Getting another color, it is gram negative. Steps, as you know, take bacterial assembly, dry it, stain with the crystal violet, gentian violet, become stain color. Wash with water, treat with iodine solution. Iodine will act as a mordant. That is, mordant means attach the dye family to the structure. So difixative, formation of chemical complex, treating with the alcohol. So usually act as a decolorizer. Those who retain violet, gram positive. Those who lost the color, counterstain with the safranin, become pink, it is gram negative. Some examples for gram positive and gram negative. Odd one question. Gram negative, remember E. coli, even though it is a symbiotic bacteria. Permanent staining is due to many reasons. One or two I given there, thickness of cell wall, murin content, etc. In both cases, more in gram positive bacteria. Still many features are there. Then we entered into next area, plasma membrane. Features, selectively permeable, originally interacts with outside, structurally similar to eukaryote. Mesosome, special membranous structure. Mesosome formed by the invagination of plasma membrane to the cell. It can exist in three forms: vesicles, tubules, and lamellae. Diagram given there, shapes as well as structure of a typical bacteria, old NCERT. Then functions of mesosome, important, connect with the cell wall formation, DNA replication, distribution, daughter cell, helps in respiration, secretion process, increases surface area, plasma membrane, enzymatic content. Next is important, mesosome is common in gram-positive bacteria. Gram-positive bacteria. Next, in some cyanobacteria, purple bacteria, some other membrane extensions also seen into cytoplasm, which is chromatophores, pigments in, helps in photosynthesis. Now comes motility of bacteria, how bacteria moves. Bacteria may be motile or non-motile, but if it is motile linked with flagella, cilia not seen, number of flagella variable. Now structure of flagellum, for that also I put old NCERT diagram, three parts, filament, hook and basal body, filament here, you can find hook here, basal body with the rings, etc. Then rings, not directly given NCERT, but remember 4 rings, LPSM, rings MS in cell membrane, PL in cell wall. You can find PL will be outside, SM will be inside, cell membrane inside. Then next aspect for us is gram negative, 4 rings, gram positive, that is 2 rings. Gram positive, 2 rings, SM, gram negative, 4 rings, LPSM. MS in cell membrane, PL in cell wall. I think that's clear because not familiar to most of students, not directly given in present NCERT. Still, I just mentioned it. The paper is a bit deep, etc. Pili, fimbriae. Besides flagella, pili and fimbriae also suffer structures in bacteria, but not connected with the motility. Pili, elongated tubular, made by protein pilin, connected with the sexual reproduction. True pili, like a mesosome we just discussed previously, commonly seen in gram-negative bacteria, sex pili or F pili. Fimbriae, small bristle-like fibers sprouting out of the cell function in some bacteria. They are known to help in attachment of bacteria to rocks in streams and host tissue. Now, inclusion bodies. Here you can find phyli, old NCERT. Ribosome and inclusion body. Prokaryotic ribosome is seen associated with the plasma membrane, size 50 nanometer to 20 nanometer, made by 2 subunit, 50, 30, total 70, main function. Protein synthesis. Several ribosomes attached to in a single mRNA forms a chain called polyribosome or polysome, or rarely a term used, ergosome. But main term, polyribosome or polysome. Ribosome of a polysome translate mRNA into protein. Now comes last part, inclusion bodies features. Rhizometal in prokaryote, stored in cytoplasm, form of inclusion body, not bound to any membrane system, free in cytoplasm. Phosphate granules, synophysing granule, glycogen granules. So main item. There are another type of inclusion body. There may be a covering, usually single layer, but it is not unit membrane, it is non-unit membrane, 2 to 4 nanometer thick. Example is sulfur granules, gas vacuoles, poly beta hydroxybutyrate granules. Gas vacuoles are seen in blue-green algae, purple and green photosynthetic bacteria. Then we entered into the last part, that is introduction. Eukaryotic cell introduction. Eu came from Greek, means true good. Karyo means nucleus, that is totality. Eukaryotic cell means cells with organized nucleus and nuclear emblem. Then as usual, 5 theory question, 5 NEET model question with the key. If you like this episode, please subscribe the channel Ace Botany. So you'll be getting daily dose NEET question also. Theory also. After finishing this theory, 3 more chapters, then we'll be taking segments and questions separately. We'll be doing it. So subscribe Ace Botany. For time being, we can wind up. Thank you. Take care. Bye. All the best.